Well, first of all, I would like to welcome everybody to this thesis defense. It's good to see so many people in the audience. And therefore, I would like to open officially this academic ceremony in which Ms. Isabeau Vermeulen will defend her academic thesis entitled Multi-omics in Drug Discovery and Development. May I invite the candidate to present a summary of your study and the conclusion of your thesis in the next 15 minutes, and I'd like to give you the word.
Thank you very much. Dear Prorector, dear jury members, dear supervisors, dear family, friends and colleagues, thank you all for being here today, where I will be presenting my PhD research entitled Multi-omics in Drug Discovery and Development. And I would like to start off with giving you a short overview of the drug development pipeline. So it all starts with target identification and validation, where researchers pinpoint a molecule that plays a key role in a disease. In the next step, we screen thousands of compounds to see which compound can interact with the target of interest. Once we selected a few candidates, we go to the optimization of those compounds, where we refine it to make it more safe and effective. To continue with preclinical studies, where we investigate the effects of the drugs in an animal model. Afterwards, we go to clinical studies, where the drug is tested in human volunteers, to eventually obtain approval and the drug can reach the market. And this process can take up to 15 years. During my PhD research, I focused on two key steps. One is target identification in epilepsy, and the other one is preclinical studies, where I looked at PET tracer distribution and the effects of new drugs on liver disease. So let's start with target identification, where I used multi-omics technologies. And that starts with transcriptomics, where we investigate the RNA. And this RNA serves as a messenger to tell the body which proteins needs to be developed. And these proteins can be measured by using proteomics. And proteins are actually the molecules that perform most of the essential functions in the body. And even we go one step further, where we investigate the metabolites and the lipids, called metabolomics and lipidomics, to see which small molecules are involved in the cellular metabolism. And one of the key technologies that I've used to investigate the lipidomics is MALDI-MSI, or matrix-assisted laser desorption ionization mass spectrometry imaging. So let me shortly explain how this technique works. So we have a tissue, and we cut that tissue into really thin sections, and we put that on a glass slide. After, we apply a chemical matrix, and this matrix allows that the molecules come loose from the tissue and can fly to the detector. So what we do is we shoot with a laser, the molecules are desorbed, they get ionized, and in this case the lipids can fly to the detector. So how does the imaging part work exactly? So the tissue is divided into pixels, just like a photograph, and then the laser can shoot in each pixel and create a spectrum, which looks like this, and the spectrum gives then the molecule with its intensity. And then if we combine those pixels with the intensity, we can create molecular heat maps showing exactly where the molecule is located in the tissue section. So this I have performed for lipidomics. However, for proteomics, the coverage is not very high. So during my PhD research, I combined MALDI-MSI with laser capture microdissection. So how does that work? We use the spatial maps of MALDI-MSI to see which part of the tissue we exactly want to cut out. So we use a laser to cut out that tissue area, and then we perform a trypsin digestion that cuts the proteins into really small fragments. And that complex mixture is then measured with liquid chromatography mass spectrometry, which also results in a spectrum where we could see then the intensity of the peptides or the protein fragments and see which proteins are changed. So the combination of MALDI-MSI and LMD I have performed in target identification in focal epilepsy. So what is focal epilepsy? It's a type of localized epilepsy, so it's only present in a part of the brain. And usually it's characterized by structural abnormalities. It's one of the most drug-resistant causes of epilepsy. So currently, the only possible treatment is surgical removal of that epilepsy zone. So in chapter 3, I focused on focal cortical dysplasia type 2, or in short, FCD type 2. And this is a type of focal epilepsy that is caused by a mutation in the mTOR pathway, which then causes an abnormal brain development that eventually results in epilepsy, and that FCD can be present in any part of the brain. In chapter 4, on the other hand, I focused on temporal lobe epilepsy, so that epilepsy is present in the temporal lobe. And in the case for surgical removal of that epilepsy zone, of course, the surgeons need to decide where the epilepsy zone is located. So they can perform a positron emission tomography scan to see where the epilepsy is located. So if you look at the top, you can see that there is a more blue region, which corresponds to less metabolism. So in epilepsy, the metabolism, the glucose metabolism is less, so they can exactly see where the epilepsy is located. However, if you look at the bottom, there are patients with temporal lobe epilepsy that do not have a change in metabolism, so you cannot see where exactly it's located. So the goal would be to investigate what the difference is in that cellular metabolism over there. So let's first look at the molecular changes in FCD type 2. So we had a mouse model and we performed multi-MSI to look at the lipidomic changes. And we saw two lipid classes, the sulfatides and the phosphatidylserines, to be downregulated. And as you can see here on the right, you can see the molecular heat maps that I was talking about, where you can clearly see where the FCD lesion is located. So then we went with a laser around that FCD lesion to look at the proteomic changes. And we could see that proteins related to myelin were downregulated, but we also found proteins involved in cell signaling, neurotransmitter balance, and immune response. So if we would combine all that data from the animal model, we could say that there is some sort of myelin disruption present in the FCD region. So in the brain, the neurons communicate through the axons and send their electrical signals through those axons. And if you look at the top, normally those axons are surrounded by a normal myelin. But if you look at the bottom, that insulation is not present there anymore. And that is what we think is happening based on our data. On top of that, we also looked at human samples of FCD, and we performed transcriptomics, and there we could see that actually the genes related to myelin were increased. So what we think is happening is that the brain is trying to compensate for the damaged myelin by increasing the production of myelin proteins. Let's move on to the molecular changes in temporal lobe epilepsy. So we started with the golden standard to look at the tissue morphology and H&E staining. We applied this on neocortex and hippocampus samples of human patients because that is exactly where the epilepsy zone was located. So we could see the different regions within our sections. On top of that, we applied multi-MSI, where we looked at the lipid profile. And then if we combined all of that data together also with proteomic analysis, we found several pathways that could explain the difference in metabolism between those patients, which you can see here on the slide. And these different pathways could help future research to direct how potential new drugs can be developed to treat temporal lobe epilepsy, for example. Now I would like to move on to the preclinical studies, and more specifically the PET tracer distribution. So some patients can undergo a positron emission tomography scan to, for example, look where a tumor is located. So they get injected with a radioactive label, and then exactly the signal shows up where a tumor is, or for example, as I showed before, where the epilepsy zone is. However, during the development of such PET tracers, there are some disadvantages. First of all, it's radioactive, so it's not really safe. Also, they have a short in vivo half-life, which means the radioactivity decays over time. So if we want to perform long-term studies, that's not really possible because we cannot detect the radioactive signal anymore. It's very expensive as well, and it's not always feasible to make a radio-labeled compound. So what we did was maybe we can change during the development to multi-MSI and look at the distribution of those PET tracers without using those radioactive labels. So how did we do that? We mixed rat brain with a well-known PET tracer. And as you can see on the left, we had different concentrations. And if you also look at the left, the goal was to have a really high signal. And with a really high signal, I mean the more yellow, the more white. So if you look at the left column, we could see that the signal was not optimal. We would want it to be higher. And this is caused by ion suppression. So during the ionization of the molecules, some molecules get ionized really easily and give off a strong signal. But there are also molecules present in the tissue, just like the PET tracers, that doesn't get easily ionized and they give off a weak signal. So the goal was to implement washing steps. So we wash the tissue with an organic solvent so that we can remove the ions with the strong signal and improve the signal of our own PET tracer. And that's exactly what we did on the right. So we applied dichloromethane and ethyl acetate. And as you can see, for the higher concentrations, the signal is more yellow, more white. So we improved the signal there a lot, and that is exactly what we wanted to obtain. Of course, we also applied this in dosed rat brains. So we had the rats dosed with that PET tracer. And here you can see the tissue sections of those rat brains. And as you can see, after an ethyl acetate wash step, the signal is clearly higher compared to the no washing. And if we would go into a bit more detail and compare it with the H&E staining on the bottom, you can see that after an ethyl acetate wash step, we do see it more in the cortex and the cerebellum, and that is exactly where we expect the tracer to be. However, if we look at dichloromethane, we see it more in the brainstem and in the corpus callosum, where it's exactly not supposed to be, so we think it's the free UCB tracer. Now let's move on to the preclinical studies, and more specifically the effects of new drugs on liver disease. And in this case, I focused on metabolic dysfunction-associated fatty liver disease, or in short, MAFLD. So because of the current lifestyle and our diet that changes a lot, this disease is becoming increasingly important and also widespread. So in the case of MAFLD, there's actually an accumulation of fat in the liver. And this can even go further to steatohepatitis, which is characterized by inflammation and fibrosis. So in chapter 6, I focused on a new potential drug that activates AMPK, and AMPK is involved in cellular metabolism. And in chapter 7, I focused on cathepsin D inhibition. So cathepsin D is actually an enzyme that is present in the lysosomes. However, during the disease of MASH, that enzyme can go outside of the cell. So in that chapter, we investigated what would be the most beneficial if we treat the liver with an intracellular cathepsin D inhibitor or with an extracellular cathepsin D inhibitor, and which one would be the most beneficial to treat that liver disease. So first of all, for the AMPK activator, we investigated the lipidomic changes in a mouse model, and there we saw that the lipid profile was a shift towards a normal composition, so a more healthy composition. We also looked at the proteomic changes, where we could see that the fatty acid synthesis was downregulated and the proteins related to fatty acid oxidation were upregulated. So if we would combine all of that data together, we could say that AMPK activation promotes a catabolic switch. And in short, that actually means that we saw a decrease in fat accumulation, which is exactly what we want in fatty liver disease. Moving on to the cathepsin D inhibition. As you can see here on the slide, this is a multi-MSI image of a cardiolipin. And as you can see, the signal after intracellular cathepsin D inhibition is similar to the control one, but in this case, the control one is a disease state. So it's actually very similar to the disease. And those cardiolipins are actually a lipid class that are involved in the production of reactive oxygen species. So reactive oxygen species are actually molecules that if they are present in the cell, they damage the cell, so we don't want them there. So our goal was to see what the presence is of those ROS species after treatment with either the intracellular or the extracellular inhibitor. And as you can see here, we tested those in cells, and you can see that the signal, the blue line for the intracellular inhibitor, definitely goes up, so there's an increase in ROS production there. If we combined all of that data together, we could actually say that the extracellular cathepsin D inhibitor is a better treatment option because there is less ROS production, there are also less cardiolipins, and even further, we also found lipid classes related to inflammation to be downregulated in the extracellular cathepsin D treatment. So overall, what I have tried to show with my PhD research is that MALDI-MSI can support target identification and preclinical studies. I have enhanced the understanding of epilepsy at a molecular level in Chapter 3 and Chapter 4. I have tried to enable imaging of PET tracer distribution in Chapter 5. And in Chapter 6 and 7, I assessed the impact of new drugs on liver disease. And with that, I would like to conclude my presentation and give the word back to the prorector.
Thank you very much, Ms. Candidate, and you're exactly on time, so that already shows the very appropriate behavior. Before we start officially the questioning of your thesis, I would like to mention to the audience that we have 5 opponents today, and one of the 5 will be online and will appear on the screen. And also, I would like to start mentioning your promotion team, because they have been involved in the last 4 years to work with you since 2019. And there are 3 members here at the promotion team, and I'd like to also mention them to you. From my right side here is Dr. Barton. He is head of the Department of Drug Metabolism and Pharmacokinetics in UCB Pharma at Leuven, I suppose. Yes, thank you. Then we have Dr. Silero Pastor. She is Associate Professor at Spatial Omics for Regenerative Medicine here in Maastricht. And of course, the first promoter here, always on my left, is Professor Heren. He is head of the Department of Molecular Imaging. And I would like to thank them that they guided you in the last 4 years. So without any further ado, I would like to start, and we're going to open the official questioning, the Corona members here present, and I'd like to give the word to Professor Honing. Professor Honing is Professor in Analytics for Systems Imaging at Maastricht University, and I'd like to give him the opportunity to discuss with you first.
Thank you, Mr. Prorector. Dear candidate, very much congratulations for this great work, pushing mass spectrometry imaging in the field of pharmaceutical sciences and trying to sell the sciences. I will come back to that. But first I have an intriguing question. There are some pages 21, 22, 75, 76. They're empty, and yesterday evening I was shining with light to see hidden letters. Is there a purpose behind this?
Highly esteemed opponent, thank you for your kind words and for your question. So I must be honest, those are in there for the layout, so that it's clear.
Oh yeah, you were not fooling me.
No, I was not fooling
you. Thank you.
Yes.
Then otherwise I would be angry with you, but okay. Now for the more serious things, I would like to ask you if one of your assistants can read proposition number 2.
MALDI-MSI offers non-radioactive alternatives that aid in decision-making in drug development studies by the quantitative visualization of PET tracer distributions.
Thank you very much. Dear candidate, these are big words, and here you speak about decision-making. What do you mean with decision-making? What decision-making?
So the goal was to show that MALDI-MSI can actually be used in the beginning stage of the PET tracer development. So if we have a certain candidates at the beginning, not radio-labeled, to look at their distribution.
But what decision? Of whom?
To see if we can choose that candidate to go to the next stage of the development phase.
And do we need radio-labeled tracers for that, or can we do just pharmacokinetics and select on the structure-activity relationships in vivo?
So in the beginning stage, we could do that. At a certain point, we would need to go to autoradiography because we would have the radio...
That's mass balance analysis, and then you make the decision if you have retention of your molecule. But how is this going to help you in decision-making between optimizing the properties of the structures of the molecules?
So I would say that in the beginning, the goal is, of course, to see if the PET tracer can reach the intended target.
Okay, so it can reach it, and then hopefully with imaging you can see that.
Exactly.
I think I'm completely agree with you that it would be very interesting, but I would also argue you cannot distinguish between an agonist or partial antagonist or even dual inhibitors.
No, I agree
Okay.
with that. I agree with that. This would be more in the initial stage, if you have a lot of candidates still, to just see if they could reach the intended target.
Yeah,
and then
maybe good to say that, especially because in chapter 6 and 7 you speak about kinases, and in kinases there's a big issue, that's selectivity. So how can you then use this where you would like to see the biological effect of selectivity in vivo, do it with your technique?
Yeah, so that's a bit a difficult thing, and that's something that still needs to be investigated as well, because during my research of those PET tracers, we used a very high concentration as well.
Yeah, that's very interesting, because you were looking to ROS. I don't know the page anymore. Doesn't matter. But you were using concentrations of 100 micromolar. That's far from what is normally, I hope, administered to me.
Yeah, so
you create chemistry which is normally not there.
So you mean for the ROS production in chapter 6?
100 micromolar.
Yeah, so for that, this was done by collaborators, and we discussed this up front, of course, if this would be like an appropriate concentration to do. We also tested this in cells, so we didn't test this
And they did not
vivo.
die?
No.
100 microliter? Okay.
Yeah.
Not very interesting. Yeah. I'm interrupting you, but I think we agree that mass spectrometry will have an impact. In pharma for sure, but we should balance a little bit on what it is promising, because you even state at the beginning of chapter 2 that you are going to help to improve the attrition rate, and apart from high throughput screening, computational chemistry, modulation, neural networks, organ on a chip even, nothing delivered in the last 20 years. So how is your technique going to deliver the increase of the success in the preclinical development line?
So I have to agree this was a pilot study. So it was it's not done a lot before. So our goal was to show at least that MALDI-MSI is able to show those PET tracers.
Then we completely agree.
I fully agree that we're not there yet. Yeah. But I think once we get to the fact that we can challenge the ion suppression, those kind of things, that we would have an influence on the attrition rate and everything.
There we agree fully.
So, yeah.
Thank you for your answer. Now I'm going to teach you a little bit more on the analytical chemistry. So on page 18, you speak about NMR. Can you tell me the abbreviation of what NMR means?
Sorry?
What does NMR mean?
Nuclear magnetic resonance.
Okay. I'm happy to hear that, because you are speaking about isotopes, and I don't think we are analyzing isotopes with NMR. And then you try to put NMR in such a position that you come up with mass spectrometry as a much better alternative, because there's a lot of peaks in your NMR, but we have two-dimensional, three-dimensional NMR, even COSIS, TOXIS, and MS will never give you any conformational space analysis of 3D structures. So just to ask you, do you prefer, and I know your professor or your supervisors are here, you have to say to me NMR is much better than mass spectrometry. Yes, Professor Honing? No?
So I think it depends kind of the question you would like to answer, if you would use NMR or if you would use mass spectrometry in that case. So it's a neutral answer to not pick sides.
Then the last small question on page 303, and then I catch you again. There's a spectrum of the ATX 304 compound, and unfortunately you did not give me the structure, but I found it, and I looked to the structure, and then I look to the MS-MS spectrum, and I see only one peak in the MS-MS. So that's rather poor to assign molecular structures in mass spectrometry with only one fragment. Do you agree with me?
So if I go to that page, I would not say it's only one structure. Fragment.
Yeah, there's two small products, but if you have a complex molecule with two chlorines, sulfur, and a lot of nitrogens, then still I'm wondering why you have only one major fragment which is not really indicative of molecular structure, especially not conformational space and so on. I think that was the discussion. I'm sorry for interrupting you, but very well answered questions. Thank you.
Thank you.
Thank you very much also for your questionings, which were quite a lot of questions, and they were answered. Thank you. We will continue, and the second opponent in this morning thesis defense will be Professor Martinez. She is Professor of Neuroinflammation and Neuropsychiatric Disorders at our university, and I'd like to give her the word.
Thank you, dear Pro-Rector. Dear candidate, I would like first to congratulate you for the nice, interesting thesis, and also to your supervision team here present as well. And as you know, I'm very interested in lipids. I think that's you knew this in advance. And I really think that lipids is the future. It has been 2 Nobel Prizes already in genetics and also in protein analysis, but no Nobel Prize yet in lipidomics, so in lipid analysis. So I think it's a very good career choice. And then looking to your thesis and the very interesting work you have done, I was really interested about the fact, or puzzled about the fact that some lipids do not get ionized well, and you cannot analyze them. So do you have perhaps some ideas how to overcome this, or things that you would like to explore technically at the technical level that you didn't have the chance to test in your thesis, or perhaps you did start testing but didn't have enough time to get a conclusion?
Highly esteemed opponent, thank you for your kind words and your question. So the goal of my thesis, specifically looking at the lipids, was to have a first idea of the lipid classes. So I would suggest for future research, for example, in chapter 3, where we look at sulfatides, specifically related to myelin, if we could implement also maybe some sort of washing steps to wash off certain lipids to improve the signal for other classes as well. On the other hand, maybe also combine it with LCMS to also have a wider coverage of the lipids. So that are 2 options that I would think. And
I was thinking, so what about the positive or the negative ionization mode? So what does it mean? So how does it could be helping to analyze certain lipids if you change the polarity of the...
So in chapter 3 we did both polarities. So we looked in positive mode to look specifically at the phosphatidylcholines, which are mostly present, like easily ionized in positive mode. We combined also that data with negative mode, where we then more see the phosphatidylinositols and sulfatides. So in that way, actually, indeed combining both polarities also increase our coverage of the...
Okay, so that could be a good start. So in order to develop a more exhaustive analysis of the lipids in the tissue, so that a recommendation will be made to perhaps have different type of settings present always, so not to be biased towards one class or another class. I think that could be interesting. So now looking to your thesis as well, you focus in epilepsy and a relevant disease, of course, and you use human material, which I think is very valuable, and very few studies doing this. But I just wonder, so on the view of your results, would you miss to have perhaps done some experiments using animal models, because animal models have versatility, gives you possibilities. So what would you have done with the results that you have achieved in your human studies? What do you miss, or would you have liked to analyze in animal models?
So for chapter 3, we only looked at the lipids and the proteins in the animal model, and we only performed transcriptomics in the human data. So it would, of course, have been the best to also look at the lipids and the proteins in the human data, and also the other way around, to look at the transcriptomic, do a transcriptomic analysis in the animal model.
But what precisely in the animal model? So, for example, when you look to your human tissue, you are looking to a disease, the disease which is quite developed already, right? So, what advantage will you have if you will use an animal model? So, what can you change in the animal model in order to analyze better what is happening?
I'm not sure if I get... So if we want to look at a certain change in lipid classes, you mean, we could work with the knockout model?
So you could, what I mean is you could work over time. So you could do a timeline or a time course of the experiment so that you could see whether there is an accumulation, a change that is correlating. So I think that could give you a lot of possibilities. Then when you look into your data, sometimes you see lipid changes, but the genes or the enzymes that are involved in the changes of the lipids are not picked up by your proteomics or transcriptomic analysis. So could you elaborate why that will be? So why is that not really a correlation?
So during multi-MSI, we usually only detect the lipid classes that easily ionize. And for the protein analysis, we have a higher coverage of the proteins. So it's not always that maybe we find the same proteins related to those lipid classes, because we have also a really tiny area. So I would say it's more a matter of ion suppression as well, that we do not detect everything. Mm-hmm. And the most difficult part also in this was we find certain lipid classes, both upregulated and downregulated. And there's not today a software available that allows me to put all my lipid data and all my protein data together in the software.
Okay. That's a good point, very good. So I was now also looking into chapter 4, also very interesting, so that you say, okay, if we will be using lipid-modifying drugs, which are very attractive and very highly unexplored, we could then analyze perhaps if our MSI will help us to identify those changes on the particular lipids that are modified by the drugs. And that's a very, and I do agree with you, by the way, and then you give a number of examples, mainly in the cancer field, but not in the brain. Well, the brain is the organ of the body that has the most lipids and also will be the best to analyze, because in fact there are many drugs tested in humans that modify lipids in brain diseases, like for example multiple sclerosis. We, for example, have worked in AD. So could you elaborate perhaps how will you design such an animal experiment if you will be targeting the brain with your MSI? Which type of perhaps of experiment would you like to perform? Will you perfuse the animals? Will you not perfuse the animals? So how will you be analyzing this material?
Yeah, so in chapter 4, the animals were perfused as well. And then we looked at, would this have an effect on the distribution of the drugs? But that specific perfusion does not cross the blood-brain barrier. So indeed, I would still go with perfusion based on discussion with the collaborators. But indeed, if we would have a new drug, I would start with an animal model and look at the location of the drug with multi-MSI and also see specifically which lipids that correspond to the distribution of the drug are also up or down regulated.
And then one small question, and then I give the word to the pro-rector. So I also, when you talk to people, experts in every area, proteomics, genomics, or lipidomics, they will tell you, okay, but your omics analysis, is this really, MSI is really exhaustive enough for us to trust this methodology? It is still in the development phase. So what do you think we need to do in order to be able to correlate all these omics in a way that is accepted by all the different fields? So what are we looking at? How many years do we need to do that?
So my idea would be, so in the beginning, to convince people to implement multi-MSI into the multi-omics workflow is also keep it as multi-omics. To not only say multi-MSI can give you all the answers, but if you combine multi-MSI together with those proteomics, together with LMD, that you have not only looking at the omics, but also have the spatial component in there, which if you would talk with the really proteomics and transcriptomics, at this current stage, they're not really into the spatial that deep. So I would say like combine it so that the spatial compound is in there. And in that way, try to improve the use of multi-MSI.
Thank you, and give the word back.
Thank you very much. Then the opposition will be continued by Professor Shi. Professor Shi is head of the Department of Polymer Therapeutics at the Uniclinic RWTH Aachen. And I would like to thank him that he joins us this morning for this defense. And I'd like to give him the word. Professor Shi, it's yours.
Thanks, Prorector. Dear candidates, it's really a great pleasure to study your thesis. It's really great to learn that you have done a lot of studies in the past 4 years, and I think this is really great work that you can be proud of. So I'm very interested in the last 2 chapters, and especially chapter 7. I think it's really a very important work that you started to look at different inhibitors for metabolic dysfunction associated steatotic liver diseases and MASH. The first question for me is that you choose 2 inhibitors. One is extracellular, one is intracellular. So the idea of this extracellular inhibitor is that it works only extracellularly. But I was trying to find more information about the inhibitors, but I couldn't find. So how sure you are that these are really extracellular and intracellular inhibitors of your target CTSD?
Highly esteemed opponent, thank you for your kind words and for your question. So the 2 inhibitors that we used in this study were already previously investigated for the proteomic changes. So I cannot say 100% sure, but in those previous studies, they did investigate, I think, the activity of cathepsin D, extracellular and intracellular, to check if it would only, but I cannot say the results of that.
Okay, so it's indeed, there was a publication on these two compounds I could find, but also in this publication, there was nothing really on the mechanistic study to show it's really extracellular and intracellular. And I think this is a really important question because this is essentially what you want to address, right? And if you think about when you claim it's an extracellular inhibitor, it means it's not taken up by cells. It's exclusively active outside of cells, and small molecules to work in this way. For me, it's really difficult, challenging. So, and then a follow-up question related to this is that if you had a chance to really look at this, what would be your best strategy to answer this question?
If I would need to see if the inhibitor exclusively is found outside or inside the cells, I think I would label them with fluorescence and add them to the cells and then see if they would be present intracellular and extracellular. I think that would give a first idea, of course, where they are located. So that would be something I would start with, and then I think some kind of activity assay to look at the difference if we treat them either with the intracellular or the extracellular and see what would be the activity of cathepsin D itself.
Great. You brought up a very interesting point, which I thought about it many times before. In my community, people often use fluorescence labels to tag biomaterials, nanoparticles, to study their in vitro behaviors. But if you use such method to study the interaction with cells of small molecules, then is it a concern that you significantly change the chemical structure of your small molecule compounds and also the physical chemical properties? For example, charge, hydrophobicity, would that be your concern?
So yeah, indeed, it could change indeed the chemical compound, but maybe if I look at it now, we can go with multi-MSI up into the single cell level. So maybe instead of using fluorescent labels, I can promote multi-MSI again for this type of research and try to see if we go to single cell multi-MSI, if we would find the drug compound outside or inside the cells.
Thanks. I think this is a really great answer to this question. I think your technique really has a much better chance to give us more reliable data than conventional fluorescence labeling of small molecules. So my second question related to chapter 7 is related to the Ross essay you did. I understood that it was probably done with collaborators, but still I would like to ask you that you are looking at a liver disease, but you choose to do the Ross assay on PBMCs. These are peripheral blood mononuclear cells. So as far as I know, immune cells are highly multifunctional. They have many different phenotypes, and especially even for the same immune cells, when they are tissue resident or when they are circulating in the bloodstream, they have very different phenotypes. So my question is that why did you choose PBMCs to study ROS in your project?
So we started the experiments with macrophages because these cells are present in the liver and are known to be involved in inflammation, etc. So of course, if we would measure the ROS presence in those cells, we would see that those cells will react to that, of course. So that was the first idea. On the other hand, of course, I agree that we would have to also measure it in hepatocytes to have a clear idea. However, due to ethical constraints, that was not possible. However, I can say that we measured the ROS activity as well in AML12 cells. That data is not in the thesis, but we also performed it in there, and there we could not see any changes in the ROS. But we also, that's the reason why we initially continued with macrophages.
Thanks. And then I would like now come to chapter 6. So here you have studied a compound called ATX304. So I suppose the target tissue of this molecule should be the liver. Is that really known?
There are previous studies with this specific compound, I think in the kidney as well. So it's not specifically to the liver only.
Okay, then do you think it is interesting for you to use your techniques to further investigate, for example, the biodistribution of these compounds in vivo?
Yes. So with this study, we could actually show that it has beneficial effect on the liver disease as a first, but it would be of interest, of course, to also include other organs to see what the effects or maybe even side effects would be on the other organs. Yes.
Okay, good. And my second question related to chapter 6 is that I understood that you started your PhD 4 years ago, before the pandemic time. And what I want to ask you is that in the last few years, in these fields, the trends or the most, shouldn't say the most important, but really attractive type of drugs for the same disease is called GLP-1. So these are now in the pharma industry. I think many companies really want to step in these fields. But your compound is completely different mechanism, different targets. So would you think it's interesting to compare your compound to GLP-1 drugs? And if yes, within your capacity, with your imaging technology, what would you study for this newer molecule?
Specifically for those liver diseases, it would be indeed for the fatty liver disease to compare also the drugs that are currently upcoming and compare those with my thesis that are not really widely spread. And for that, I would look at the lipids first, because we're looking, of course, at fatty liver disease. So in a first stage, I would compare these drugs and look at the lipid droplets in the liver itself to compare how both drugs react to that.
Mm-hmm. Apart from the lipid accumulation, would you think of some other important aspects to investigate?
So of course, I can go as broad as I want with that, but of course the proteins, looking at the proteins as well, and the RNA would also be interesting, and really use a multi-omics approach to look at the differences and how the mechanism differ between the two drugs.
Good. Thanks. Director, I finished my questions. Thanks.
Thank you very much, Professor Shi. Much appreciated. Then we continue the opposition with Dr. Lennartz. She is affiliated with the Department of Surgery at our university, and I'd like to give her the word.
Okay, and thank you, Prorector. Dear candidate, I read your thesis with pleasure, and I want to congratulate you on that, and together with your supervisory team. I am more into biology. My expertise is more in biology. So I'm wondering, chapter 6, I want to discuss a bit with you the biological impact. So these mice that you fed this drug to reduce the phenotype in the liver, you see that there is a strong drop in their body weight directly after the starting of the treatment. And you claim that the results that you found are not related to this drop in body weight and this drop in food intake. How sure are you of that? And could you maybe control for that? Do you have ideas how you could make a control group to compare?
Highly esteemed opponent, thank you for your kind words and your question. So indeed, after treatment, you see initially an decrease in the body weight, although they did not see, I think, a difference in the type of food. So I think to control that, I would control the amount of food that they are given, of course, to basically say in the end of, look, if we compare with the control group that had the exact same amount of food, we still see a decrease in the body weight. So I think in this study, even after a while, you could see that even the food intake was higher for the treatment, which also kind of shows that it's not related, like that it's specifically the drug itself that changes that.
But you see this drop is it's like 25% in 2 weeks, for instance, and their food intake is also 20% less during that period. So yeah, I would add some control groups to really see that your effects that you find are not related at all to this drop in food intake. Mm-hmm. Do you know why this food intake was reduced so much?
I think after the treatment, just because of the taste and the color, I think there were some changes in the food intake.
Okay,
yes.
So especially taste. Okay, thank you for that. Then I would like to go a bit more in, you combined, or you did actually proteomics and RNA-seq analysis, as well as lipidomics. So why didn't you choose to do these, generate these data at the same time points? I see that your protein data were generated at 45 weeks, while your RNA-seq data were of 31 weeks. So why not do all at the same time, and then you have like a more complete picture on one time point? What was your motivation to not do that?
That is a very good question. I think initially the study was designed to, I think, only do RNA sequencing, and then based on the results that were found, to continue with additional experiments. So I think that is one of the reasons why the timing are not 100% the same. While I do agree that for future research, we should have one time point to look at it and compare those better.
Okay, thank you. And then I'm interested in the differences that you saw between the left and the right lobe. So, and apparently you do not see these differences back in your data that you generated with MSI, but also the proteomics data. And then you decide to combine the data again. So you see this phenotypically very different. So what makes you then decide to just merge then again the data, and is the spatial component important? Maybe you don't need this in your,
So for the multi-MSI lipid data, so initially we did not see really any spatial distribution that obvious. So indeed the spatial component in this case, the goal was to initially see the lipid droplets, but we were not able to detect them with the lipids that multi-MSI can detect. Mm-hmm. So that's one of the reasons, because multi-MSI was not able potentially to detect those lipids involved in the spatial component. That was one of the reasons why we decided to combine those. And also the difference that we found were not that obvious between the left and the right lobe based on the limited amount of proteins and lipids that were detected.
Mm-hmm. What does that indicate? Is your data, is your method maybe not sensitive enough to pick up differences or yeah, I don't know.
So that does indicate that probably the changes are more related to the lipids that have a lower concentrations or less easily ionized, probably, compared to the ones that we detect. Yeah, that's, I think, one of them.
Could be a reason. Huh? Yeah. So then I would like to hear your ideas about, you have this different data also in this chapter, but you really describe them as separate, and I do not see the helicopter view, so really the integration of all the data that you gathered to give a complete understanding. So what would you need for that to really make that picture and to try to understand completely? You have the metabolites, you have the proteins, you have the RNA data. Yeah.
Yeah, so as I mentioned in my discussion, there is a huge need for a software that is able to have a lipid database that makes it possible to put in the lipids that we detect, that those can be combined with the proteins, that even they can be combined with the RNA data to show exactly in the biology what is exactly changing. I know it's not easy because, yeah, it's different pathways and it will require a lot of data, but I think that's one of the main things, or at least if we can combine the lipid and the protein data into one software. I do know it exists, but it's not yet at the level that was useful for my...
Okay, and I was wondering about you see as a strong increase in bile acids and plasma in these treated animals only. Did you look, because you have all your transcriptome data, we know that these bile acids, that they have strong effect also on the liver itself and they can induce certain targets. Did you look in your RNA data if there is a link between these observation in plasma and the changes that you see in your transcriptome data?
I think for the bile acids specifically, we did not look into the differences.
Because it would be very interesting to look at FXR targets, TGR5. I think you have a lot of data to also look into that in a targeted way, so and find out more about the biology. So I think I thank you for your answers and give back the word to the prorector.
Thank you very much. Then the opposition will be continued by Dr. Kuipers. She is involved in the Institute M4I here at Maastricht University, and I'd like to give her the word as well, and also thank her that she was my secretary today. Thank you very much.
Thank you, Prorector. Dear candidate, also from me, a big congratulations on the valuable work. I think it's of great interest in the field, so we will probably read the thesis a few times in the future too. So congrats for that. Of course, I also have a few questions, starting with the introduction. You also nicely showed it on the slides. The drug discovery and development pipeline, you nicely gave a few examples in your work. Do you also see added value of MSI in other parts of the pipeline, and where do you see it?
Highly esteemed opponent, thank you for your kind words and your question. So I only showed it now in 2 key steps, but I do agree that for the others it could also be beneficial. So for example, if we would look at screening of those compounds, I think if we would have some grids with the intended target on it, and then we apply different kind of drugs on top of these grids, and we could image those to see if there would be an effect. So that's one of the things that I think for screening of compounds as well. However, the same for lead optimization. If we refine certain compounds because they don't reach the target properly, with multi-MSI afterwards, we could check if they reach the target. And I think, yeah, for clinical studies, it's a bit more difficult for human material to get, so I'm not really 100% sure how unless like in chapter 4 where during surgery we would get the tissue to analyze, but then we would go back to more target identification and discovery, I think.
Okay, I totally agree with that. Thank you. Maybe also looking at the therapeutic agents and knowing you have a pharmaceutical background, you describe in the introduction drug-like small molecules or biological therapeutics. What would be your focus? What would you like the most? One of those, small molecules or biological therapeutics, and why?
That's a very good question. I think for now, I would say that small molecules would be most of interest, as I'm not saying they're easier to develop, but there are much more complex interactions related to biological agents in that case. So for small molecules, I think you can tweak and refine them a bit more compared to the other ones. So that, I think, would be my focus.
Okay, what do you mean with small molecules are much more complex?
No, I mean that like if you would go for antibodies, and those are more complex compared to small molecules, because we get them from also human material and stuff. So yeah, dosing those to humans as well adds extra complexity. Compared to small molecules, of course, it's also a chemical compound, but it does not add the fact the same as antibodies. That's what I'm trying to say.
Then maybe on chapter 3, on the epilepsy, you nicely described some lipidomic changes. Can you correlate them to the severity or the frequency of the seizures? Did you have a look to that?
So for that study, we only had like the animal model where they saw like a lot of seizures. So it's only measured at one time point in that case. However, what we could say is that because we saw the decrease of the lipids related to myelin, and also the proteins were downregulated related to myelin, but the genes, on the other hand, were upregulated, that by maturation of the brain and also continuously in the disease, that actually the myelin gets worse, and the brain is trying to compensate for that. So that's kind of a way that we could, not 100%, but could hypothesize that based on the maturation of the brain, the epilepsy, like the myelin disruption in the epilepsy, got worse.
So does that also mean that you will have changes in concentrations of the lipids that are related to the severity, or will it be ratio changes, or is it an on-off principle? What do you expect?
I think it's related to concentration as well, because of course you have the seizures as well that disrupt the insulation as well. So I think for sure the concentration will change as well. I don't think it's an on-off principle, as the genes on the other hand try to compensate for it. So if it would be on-off, I think there just would be no upregulation of the genes as well.
Okay, yeah. Makes sense. And then something to connect chapter 3 and 4. In 4, you say lipidomic differences in hippocampus and neocortex. You also use partly human samples. And there you say we did an age and tissue matching. How did you do the tissue matching, and did you take into account then this lipidomic changes in hippocampus versus neocortex? How do you deal with that?
First of all, for that study, there were only a few samples available. So we had to work with the human samples that were available. Then we performed a sectioning of the neocortex and the hippocampus. We first did the H&E staining that was annotated by a pathologist to really look like, okay, which regions are present in each section. And then we compared only the anatomical structures within each other. And then only the neocortical layers of each sample were compared to look at the lipid differences. Yeah. And also when we cut out with LMD, we only took those areas to make sure that we don't have different regions in one analysis for proteomics, for example.
Yeah, okay, that's clear. And then maybe a question on chapter 5. How would you do it for other tracer targets? Do you need to optimize again the whole protocol and the washing steps, and Or do you think you can translate it? And if so, how would you do it? Maybe with AI, or I don't know.
Yeah, so for this chapter, we used 2 pet tracers, actually, but we only looked at the distribution in dosed of 1 pet tracer. So first of all, to prove that this also works for the other, I would also apply it to the other one, pet tracer, because in this case, we used a pet tracer that was actually present all over the brain, and the other pet tracer would really only pop up in certain areas. So that's the first thing. Yeah. However, based on the homogeneous experiments that we performed, they looked quite similar. Okay. So, and we chose 2 pet tracers with different structures as well. So my first goal would still try to use the same washing step, yeah. And see. It's just 30 seconds. We try to keep it as simple as possible and also as fast as possible to make it sure that it adds up to not radio labeling the compound. So okay. That would be my first step.
Okay, that's good to know. And then can I ask one of your paranyms to read proposition 6?
As we continue to refine and improve omics techniques, the prospect of personalized medicine tailored to individual cellular signatures becomes increasingly tangible.
Of course, I had to ask a question about the cellular proposition. So, personalized medicine tailored to individual cellular signatures. How do you see that, and how would you do that in practice?
If we would look at chapter 4, where I combined like normal metabolism versus hypometabolism, that are actually 2 patient subtypes. So if we would be able to not only look at the differences overall, but also look at a single cell level, and we could use that to generate some sort of recognition pattern.
Dear Ms. Candidate, you are allowed to finish your answer, but you don't have to. You also can say, I'll finish.
Well, if we could develop some kind of recognition pattern that we can use in the clinic so that we kind of already know if it's normal or hypo, that would already help it a lot forward.
Okay, thank you.
Thank you very much. Dear Ms. Candidate, dear Ms. Vermeulen, the time appointed for defending your thesis has now passed. The degree committee will now withdraw to discuss not so much the quality of your thesis that has been approved, but more the quality, the way you have defended your thesis this morning. And I request that you and your company here in this audience await the results of our deliberations and our return to the aula. Thank you very much.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Make the mileage, traveling so far from home, gathering knowledge. and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Ms. Vermeulen, the degree committee here present has assessed the quality of your thesis, but in particular the way you have defended your thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Welcome. Professor Heren is authorized to confer upon you this academic distinction in accordance with Dutch university custom, and I invite your supervisor now to take the floor. The word is to Professor Heren.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Isabeau Vermeulen, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
I will give the word to Professor Heren, who will speak out the laudatio officially.
Thank you, Mr. Pro-Rector. Zeer geleerde Dr. Vermeulen, beste Isabeau, het zit erop. It's done. Today marks the end of a multidisciplinary journey that started a little more than 5 years ago. It was a journey that I would characterize as an adventure to the proverbial Schengen area of analytical sciences. I use this analogy as you've crossed many boundaries along the way without being limited by physical borders or passports, and you've embraced different cultures as you made your way through the different landscape where research is being performed. Your project started off with a new public-private partnership between MFRI and UCB to explore the multi-omics landscape of pharmaceutical sciences with innovative imaging approaches that we discussed today. The project was initiated by Shane Ellis and Emery Isin, who both changed their respective positions in the middle of your first year of your PhD. Their employments, they moved to different employments. It's an example of one of the unexpected challenges a traveler can experience. And the way you dealt with it demonstrated early on how independent and committed you were to the project and your success. Challenge you've all overcome. A global pandemic, instruments that break down, reviewers that are less responsive, I can go on and on. But you have overcome all of these seemingly high mountains and done so with a passion. With your two co-promoters today, Bertha Schlierer-Pastor and Patrick Barton, taking over the supervisory roles in the project after that first year, there was no stopping you at any time. You pushed forward. I sometimes actually thought that you liked those dynamics and all the new interactions along the way. Very quickly, you were achieving the first results, already after 3 months, and everybody was incredibly impressed. And it was a testimony of your eagerness to learn new technologies and understand the big questions asked by the UCB team. I remember very early on being impressed with your drive and, again, your independence. That has never changed. One of the most interesting boundaries that you've tackled or crossed was actually the interface between the interest of a pharmaceutical company and the interest of an academic research institute. While these interests and objectives align very well within your project's definition, it was you who had to perform in two very different cultures. I never stopped being impressed how you managed to bring people in the project together, explain your progress, develop the ideas, and obtain a broad support for your research in both organizations, like a true scientific polyglot. It was again a testimony of your unique drive and independence again. We all agreed that you were probably one of the most independent and hardworking PhD students we had the privilege to supervise. And the good news was we had a lot of fun along the way as well. So it was a pleasure. Whether it was at the summer school in Dubrovnik or sharing a very interesting apartment in Montreal with a big group of people, where the apartment was slightly slanting, it was a little noisy every now and then, you were always positively engaging with your fellow travelers. I want to thank you for your positive energy, even at times when not everything was going the way we wanted. But it's that unpredictable side of experimental science too. It never stopped you from keeping on pushing. To me, that's one of the true elements that has defined you as the outstanding scientist that you are. One of your unique characteristics, and we've heard a lot about that today also in the committee's evaluation, was your drive to broaden your horizon, as travelers typically do, by tackling many different projects when your own projects were not progressing as fast as you wanted. And that happened on a few occasions. Next to your main UCB projects, you also engage in projects with the neurosurgery department, the genetics department at this university, as well as UMIA University. Clearly, your planning and managerial skills have rapidly developed and will keep you moving forwards towards many new destinations. I've always been impressed with your mobility as a traveler. Whether it were your daily border crossings between Belgium and the Netherlands, you decided not to move to Maastricht, but to stay living in Belgium during your PhD, so a lot of traveling involved. Your trips to UCB, the conferences you attended, and your ability to move technologies, methods, and people forward. You're a true team player, and that sometimes needs to be reminded as well, that science is not about the individual, but science is really making things happen as a team. And that you are a true team player is also evident by the many researchers you managed to engage in your project, ranging from Joël, Nathalie, and Delphine at UCB, and many more UCB collaborators. And from this point in time, I also would like to thank the UCB team, and in particular your co-promoter Patrick Barton. Patrick, thank you very much for everything UCB has meant for this project and for Isabeau's defense. And I'm looking forward to exploring new potential future collaborations with the UCB team. So Isabeau, you also engage with many researchers at M4I and at Maastricht University that you convinced to help you and your project forward. All a testimony of the pleasant and driven person you are to work with, and a force to be reckoned with. During your PhD, you also received recognition for your work from the broader mass spectrometry community. The Dutch Society awarded you the prestigious NVMS Travel Award, and FEMS awarded you the Empowerment FEMS Award as well. Again, you keep on going places. This journey now has ended, and you start a new adventure. You left the Schengen area and moved to the UK, working in part with EMRA again, as you did in your first year, but now as a postdoctoral research associate at the University of Manchester, in again a collaborative project that suits you very well. I have no doubts that your unique drive will keep on propelling you forward in this new adventure and allow you to reach even greater heights. You've contributed tremendously to the development not only of yourself and your research, but also as M4I, bringing a positive attitude that made it a privilege and a pleasure to supervise you. I've enjoyed every minute of our joint journey together and want to thank you for everything you've brought to the Institute and the team during your M4I years. So to conclude, Dr. Vermeulen, congratulations once again with this outstanding achievement today. I'm extremely proud of your achievements during our joint journey and your success. I also want to congratulate your parents, because I think they are as proud of your achievements today as I am. So congratulations on this newly achieved honor. I'm convinced you're going to do great things in science, and you can overcome any challenge that you encounter. And with that, congratulations, and I'll give back the word to the prorector.
Dear Dr. Vermeulen, how does that feel? That feels really good. I'm pleased to hear that. I would like to congratulate you also on the board of directors of our university. You did a very good job, and I would like to mention that in our deliberations, We enjoyed, and it was mentioned, your presentation, but also the way you defended your presentation, and we came to a unanimous decision. There was not one negative voice to hear, and you really did a good job. I also noticed, but that's a small detail, I think. If you look to your slide, which is above you, you can see the green pills there, which matches with your clothing. And I think it was deliberately chosen by this. Is that correct? That's correct. So I made the right observations. Okay. So in my congratulations, I also do the congratulations to your promotion team, who brought you, as far as I notice now, officially in 4 years and 1 month to your final acceptance of the thesis by the committee. I would like to thank also the members of the Corona, who were indeed, was already mentioned by your first promoter, Professor Heren. They indeed took the time to come here and to discuss with you in detail. And again, I'd like to thank Professor Xi that he was joining us today online from Aachen. I would like to also congratulate your family here on the first line. Well, your daughter, friend, whatever it is, did a great job. And I say this is a good opportunity to also notice other students how to do a thesis defense. Compliments. I would like to come to a conclusion, and the conclusion will be that I use officially my hammer, which you haven't seen, but I'll show you right over here. And I close officially this session, and I would like to mention how we're going to proceed. We are going to, the members here of the Corona, and of course the new young doctor will stay here. We make a picture together with Professor Xi online. Then we make a picture on the stairs of our university. But you don't have to wait for this. There is already coffee, tea, even a beer and a glass of wine will be served in the rafter, and you can go straight there, and we will follow you in about 7 minutes. That's the idea. Thank you very much.